Hello, welcome to Biology, the scientific study of life. Nephrons and associated blood vessels are the functional units of the mammalian kidney. The excretory system of mammals centers on the kidneys or kidneys, which are also the principal site of water balance and salt regulation. Mammals have a pair of kidneys, each kidney being shaped and about 10 cm long. In humans, is supplied with the blood by a renal artery and drained by a renal vein. Blood flow through the kidneys is voluminous. In humans, the kidneys account for less than 1% of body weight, but they receive about 20% of resting cardiac output. Urine exists each kidney through a duct called the ureter, and both ureters drain into a common urinary bladder during urination. Urine is expelled from the urinary bladder through a tube called the urethra, which empties to the outside near the vagina in females or through the penis in males. Sphincter muscles near the junction of the urethra and the bladder, which are under the nervous system, control and regulate urination. Structure and function of the nephron and associated structures. The mammalian kidney has two distinct regions, an outer renal cortex and an inner renal medulla. Packing both regions are microscopic excretory tubules and their associated blood vessels. The nephron, the functional unit of the vertebrate kidney, consists of a single long tubule and a ball of capillaries called the glomerulus. The blend end of the tubule forms a cup-shaped swelling called a Bowman's capsule, which surrounds the glomerulus. Each human kidney contains about 1 million nephrons with a total tubule length of 20 or 80 kilometers. Filtration of the blood. Filtration occurs as blood pressure forces fluid from the blood in the glomerulus into the lumen of Bowman's capsule. The porous capillaries, along with specialized cells of the capsule called podocytes, are permeable to water and small solutes, but not to blood cells or large molecules such as plasma proteins. Filtration of small molecules is non-selective, and the filtrate in Bowman's capsule contains salts, glucose, amino acids, and vitamins, nitrogen wastes such as urea, and other small molecules, a mixture that mirrors the concentrations of these substances in blood plasma. Pathway of the filtrate. From Bowman's capsule, the filtrate passes through three regions of the nephron: the proximal tubule, the loba hindley, a hairpin turn with a descending limb and an ascending limb, and the distal tubule. The distal tubule empties into a collecting duct which receives processed filtrate from many nephrons. This filtrate flows from the many collecting ducts of the kidney into the renal pelvis, which is drained by the ureter. In the human kidney, approximately 80% of the nephrons, the cortical nephrons, have reduced lobes of Helene and are almost entirely confined to the renal cortex. The other 20% of juxtamedullary nephrons have well-developed lobes that extend deeply into the renal medulla Only mammals and bears have juxtamedullary nephrons. The nephrons of other vertebrates, like labs of Hinile, it is the juxtamedullary nephrons that enable mammals to produce urine that is hyperosmotic to body fluids, an adaptation that is extremely important for water conservation. Closer look to the mammalian excretory system. The nephron and the collecting ducts are lined by a transport epithelium that bruises the filtrate to form the urine. One of these epithelium's most important tasks is reabsorption of solutes and water. Between 1,100 and 2,000 liters of blood flows through a pair of human kidneys each day, a volume about 275 times the total volume of blood in the body. From this enormous traffic of blood, the nephrons and collecting ducts produce about 180 liters of initial filtrate, equivalent to 2 or 3 times the body weight of an average person. Of this, nearly all of the sugar, vitamins, and other organic nutrients, and about 99% of the water are reabsorbed into the blood, leaving only about 1.5 liters of urine to be voided. Notice the closer look. Excretory organs and major associated blood vessels. Notice that posterior vena cava, and this represent and renal aorta and vein. Aorta, notice that represent this one yellow aorta, and ureter and urinary blood. This is represents urethra, this one. Okay. Closer look to the kidney. Notice that. Closer look. Kidney structure. And this is the real photo. And this one, as shown here, represent renal medulla and renal cortex, renal pelvis. Renal section of kidney from a rat. This one. Notice also closer look to the renal medulla. Notice that this is the structure and function also. This one represents cortical nephron. These are, and this one renal cortex, or this one, or this line renal cortex, renal medulla. And notice the yellow also, juxtamedullary nephron, juxtamedullary nephron. And notice also collecting duct and to renal pelvis, nephron. Closer look to nephron. Notice that filtrate, the function, or the filtration and blood flow. Notice the function here is so clear in this diagram. And this is the real photo. Notice afferent arteriole from renal artery, and notice the glomerulus and Bowman's capsule, proximal tubule and pretubular capillaries, and notice distal tubule and collecting ducts. This is the line. And notice also branch of renal vein and descending limb, ascending limb, and visa recta here. Notice that, and lab of Henle. These are the function. Okay, thanks for listening.